Imagine a world where your genes are not the only blueprint of who you are. Welcome to the fascinating realm of epigenetics. Epigenetics is the study of heritable changes in gene function that do not involve changes to the DNA sequence itself. The term epigenetics comes from the Greek prefix epi, meaning over, outside of, around, indicating factors that are on top of traditional genetics. These changes can be influenced by environmental factors, lifestyle, and even experiences, affecting how genes are expressed. One of the key mechanisms in epigenetics is DNA methylation, where methyl groups are added to DNA, altering gene expression without changing the sequence. Histone modification is another mechanism, where chemical changes to histone proteins impact how tightly DNA is wound around them, influencing gene activity. Non-coding RNA sequences also play a crucial role in regulating gene expression, adding another layer of complexity. These epigenetic changes can persist through cell divisions and even be passed down to future generations. A striking example of epigenetics in action is cellular differentiation, where a single fertilized egg develops into various cell types, like neurons and muscle cells. This process involves activating some genes while silencing others, guided by epigenetic mechanisms. The concept of epigenetics has evolved over time, with its modern understanding emerging in the 1990s. British embryologist C.H. Waddington coined the term epigenetics in 1942, envisioning an epigenetic landscape to describe cell fate determination. Waddington's metaphor of a marble rolling down a landscape illustrates how cells become increasingly specialized. Recent advances have formalized Waddington's ideas, using systems dynamics to study cell fate and predict developmental outcomes. Epigenetic changes are not always permanent. They can be reversible, adding a dynamic aspect to gene regulation. For instance, histone acetylation can loosen DNA, making it more accessible for transcription, while deacetylation tightens it, repressing gene activity. DNA methylation often occurs at CpG sites, where cytosine is converted to 5-methylcytosine, impacting gene expression. Methylated cytosines in promoter regions usually repress genes, while those in gene bodies can enhance expression. Epigenetic mechanisms also include the action of repressor proteins that bind to silencer regions of DNA, further controlling gene activity. The interplay between DNA methylation and histone modification creates a complex regulatory network. For example, methyl-binding domain proteins can attract histone methyltransferases, linking DNA and histone methylation. Epigenetic changes can be influenced by various factors, including diet, stress, and environmental exposures. Foods like soy isoflavones and bilberry extracts have been shown to impact DNA methylation and oxidative stress markers. Epigenetic alterations can also occur in response to DNA damage, with repair processes sometimes leaving lasting epigenetic marks. In the brain, epigenetic changes play a crucial role in memory formation and maintenance, affecting gene transcription in neurons. Strong memories can trigger DNA double-strand breaks, leading to the activation of immediate early genes essential for memory. Epigenetic mechanisms are also implicated in brain aging and cognitive decline, highlighting their importance in neurodegenerative diseases. Epigenetic regulation is dynamic and reversible, offering potential therapeutic targets for various conditions. The study of epigenetics extends beyond humans, with significant implications for plants, animals, and even microorganisms. In bacteria, epigenetic mechanisms like DNA adenine methylation play roles in virulence, cell cycle regulation, and gene expression. Epigenetics offers a new perspective on inheritance, evolution, and adaptation, challenging traditional genetic paradigms. Stay curious and explore the world of epigenetics, where the environment, experiences, and even your choices can shape your genetic destiny.